do we use for patients who come to this hospital with AFM? And if you visit the CDC's guidance on their website about this, you will see the sentence in that green box. There is not enough human data to support preferred use or avoidance of any of the following treatments. So we don't have evidence to guide what it is that we should be doing. Some of the strategies that have been used in the past are either on the immune system modulation arm. So there was mention of intravenous immunoglobulin earlier, which was promising in a mouse model, and we have certainly used that in human patients before. Steroids actually might be harmful in a mouse model, so it's difficult to know whether or not we should avoid those. And other things like plasma exchange, where you remove a patient's antibodies, or other immunosuppressives or biologics exist and potentially could be used if we thought they might be helpful. And then on the other side of things, we have medicines that work against viruses, so antivirals. There was actually some in vitro data showing that fluoxetine or Prozac, which is a antidepressive medication, had some in the dish activity against enteroviruses. So this was actually a drug that was tried in 2018 when we had patients with AFM in an off-label way. And actually some work that Kevin was involved in put together a really beautiful case series of these patients and unfortunately showed that we didn't see as much benefit as we maybe would have hoped. There are actually no currently available antivirals that target EBD68, and there is not good evidence for interferon either. So as stepping into the lab side of things, it's really important to understand what the mechanism is that's driving paralysis so that we can really focus what side of the treatment we need to be on. So what do we have so far in the field as far as models for EBD68-driven CNS disease? So I would say that probably the best mouse model that is really able to show paralysis, because mice themselves are not native hosts for enteroviruses, is a neonatal mouse model where mice are intracerebrally injected with EBD68 and reliably go on to develop one or multiple limb paralysis. And you can see, if I get my pointer here. So in this microscopy image from the cervical spine of an infected mouse, the pink staining that you see here are neurons, and the green staining is virally infected neurons. And you can see that they tend to coalesce or co-localize in that merge image there. There has also been some work with a respiratory model that Dr. Ruckert mentioned earlier, where there is a mouse-adapted virus that can cause disease in mice. And this was first described in an IFNAR background mouse, so a mouse with an immune system that's not quite right. And there's also been some work in murine brain slices that has shown that we can apply EBD68 to brain slices of a mouse and get some infection in neurons. And then when we move to human models, there has been a cell line model. So there's a cell line called SHSY5Y that is able to be infected by neurotropic, but not non-neurotropic strains of EBD68. And finally, human-induced pluripotent stem cells, which I'll talk a lot more about today, can be encouraged to differentiate into neurons, and those can be infected with neurotropic EBD68. So that is the data that existed when we began, and we set out to create a multicellular human model for EBD68 spinal cord infection, which comes in the form of an organoid. So for those of you who are not familiar with organoids, it is a word that has been created to describe a three-dimensional model system that has multiple cell types that resembles an organ, so an organoid. And we can use induced pluripotent stem cells in the lab, and over a period of 2 or 3 weeks, apply a series of growth factors to encourage them to differentiate down a spinal cord lineage. So I have 2 different protocols or recipes that I will show today. So this one is called the SMN condition, or the spinal motor neuron. And what you're seeing in these pictures is over their development, day 1, day 4, day 7, all the way to just by eye here at day 21, you can see that they sort of coalesce into a structure and then develop over time until ultimately they're generously about a cubic millimeter. And they tend to be a little bit pleomorphic in shape. And this condition in particular, the SMN, is made mostly of spinal motor neurons. But the paper that originally described these organoids also did a different condition that actually involved fewer growth factors in the beginning. So by taking away some of those signals, it actually encourages the development of additional cell types. So you can see that they look fairly similar, both under the microscope and by eye. This is the three-disc condition, or three-dimensional spinal cord. And this actually has more cell types. So this one has spinal motor neurons, but also has interneurons and some glial cells. And we can characterize that these conditions are different from one another by using our knowledge of the neural tube. So when humans develop, the spinal cord starts as a tube called the neural tube, and there is a gradient of signals that happens along that tube that then allow for the differentiation of different parts of the spinal cord. So we can use our knowledge of what those genes are to see that in our spinal motor neuron condition, we have an enrichment in OLIG2 and NKX 6.1, which are both from this motor plate area. And then pretty similar expression of PAX6, which is this green bar that's expressed all along the neural tube. But then in the three-disc condition, we have more expression of some of the more dorsal markers, so things like PAX7 or OLIG3, and even this roof plate marker, LMX1. So we see that the three-disc condition has markers that are spread more broadly along the neural tube, while the SMN condition is really just focused on that motor neuron region. So to characterize these organoids, we wanted to assess their viability over time so that we could plan our experiments well. And we learned that cell numbers stay relatively constant and we have high viability through at least 28 days post-differentiation. And after we seed them, we end up having about 1 million cells per structure. And then we did notice that there is a little bit of a drop in cellular viability if we go out to 35 days post-differentiation. This is probably because, unfortunately, there's not yet like a vein to the center of the organoid that can deliver nutrients. So you do get a little bit of necrosis in the center if the structures get too large. So whenever you're trying to develop a model system for a viral infection, it is important to see if that virus actually does infect the model system that you are trying to develop. So that was one of the first experiments that we wanted to do. The first data that I'm showing you is in the spinal motor neuron condition. We infected the spinal motor neuron organoids with EVD68, a neurotropic strain from 2014. And at 48 hours post-infection, evaluated them for the presence of viral RNA, and we saw a really nice signal there. We also did this with immunofluorescent microscopy. So what you're looking at here, this is a mock-infected organoid. The blue is the nuclei of the organoid, so many, many cells. And the green that you're seeing is a type of neural tubulin. And if you see any red staining, that's the viral signal for EVD68. So when we zoom in here, this is just an example of the mock again. And then in this organoid that was infected with 2014 isolate, you can see some red staining here along the periphery, which was very encouraging that these were able to be infected. And then we saw something very similar with the three-disc condition that has multiple cellular types. So in these, again, we could detect RNA signal when we infected organoids over a course of 48 hours. And something that I thought was interesting is that the tubulin actually looks much more homogeneous in these particular structures. So the distribution is a little bit different, but they do seem to be just as infectable. And you can see some red staining over here in the infected organoid. So we've talked a little bit about clades today, but haven't gone into, I guess, a ton of detail. We did get a little shout out to Freeman, the prototypical strain that is from 1962, so has been richly passaged in cell culture for over 60 years now. But we often include that in our work just because we know that Freeman is not a neurotropic strain, respiratory disease only. So that's a helpful comparison. So the virus that I just showed you was one from this A2 clade. And we'll come back to some of these other clades in another slide or two. So in addition to seeing RNA signal and seeing fluorescent microscopy, we wanted to be able to show, are these organoids actually productively infected? Are they able to accept a virus into a cell, create new virions and actually release titer into the supernatant. So we did that using a growth curve. And what we learned is that the AFM-associated 2014 isolate, but not Freeman from the 60s, is able to cause productive infection in these organoids. So what you're looking at here is a viral growth curve. So we've got titer on the y-axis and then time up through 2 weeks on the x. The pink squares here at the bottom are firm. We didn't detect titer at all. And then the blue triangles are the 2014 isolate that does seem to be neurotropic. And we had a modest but consistent amount of titer throughout that entirety of that experiment. One of the things that I thought was really striking is, so as we are just visually looking at these organoids under the microscope during this 2-week period, if I covered up the labels and didn't know what I was looking at morphologically, I wouldn't be able to tell that they're infected. They really maintain their structure, their structural integrity. These images go out to 96 hours post-infection. And the top row here is mock. And I think if you compare Freeman or the neurotropic strain, you'll see that they look very similar. And then we were able to quantify that as well and did not really detect a meaningful difference. So what that said to me is there's probably not an incredible amount of cellular death that's happening during the course of this infection. So if we look at a couple of the other clades, because I've really focused just on this clade A2 so far, I wanted to know what we saw as we kind of spread across some of the other clades as well. And just to point out kind of some important dates, so 2014, this vertical line here, is when the CDC began tracking AFM. And then another important date that actually was brought up once today already is the New England Journal case where there was EBD68 staining in the spinal cord of a boy that died with AFM was actually from 2008. So that's kind of the earliest evidence that I would say that we have of this neurotropic transition, but we don't have a ton of viral isolates from that time. So I also included a 2009 isolate just to see if we could help elucidate when that transition may have been. So I have a lot of microscopy to show you here, but essentially you're again looking at infected organoids at 24 hours post-infection. This is the condition that has the multiple cell types. You're looking at blue nuclei and red actin. And then if you see any yellow, that'll be viral staining. So we do not see significant viral signal in our mock condition, our Freeman condition, or in that virus from 2009. But what we do see is lots of viral signal in many of the other isolates that emerged 2014 or after. So we've got representatives from all of the clades except for clade C, because those are not widely available, that show that they do have the capacity to infect these neural organoids. And this is also recapitulated in the titer data that we see. So any of those organoids that you saw that were infected in the last slide also had measurable titer, some even to a higher degree than the original titer data that I showed you. You know, I'm running a little bit short on time, but I do often get asked if the virus is just adherent to the surface of a sticky structure. And it turns out that infection actually goes beyond surface level. So as we zoom through a Z-stack, we're able to see some depth of infection. And I have a movie that hopefully will show that as well. There you go. So any yellow that you're seeing is virally infected cells. And then we start to be limited by the penetration of light. So there's not much I can do about that. Okay, and then just as I'm wrapping up, we did also learn that comparatively, a much more lytic enterovirus that I know is echovirus 11. And I want to point out that when you look at these echovirus 11 infected organoids at 24 and 48 hours, they're completely destroyed, right? They look like they've been through a shredder, which is very different than what we were seeing morphologically with EBD68 infection. So I did do a bit of investigation into the apoptosis pathway, which is only one pathway of cellular death, but we did some caspase-3 staining to look at an early marker of apoptosis. And what we ultimately learned is that EBD68 infected organoids do induce some degree of apoptosis, but it is significantly less than that induced by echovirus 11. So there is some cell death, but it's probably not the predominant thing that's happening inside the organoids. And then we also looked at a later pathway member with tunnel, which labels DNA breaks. And we saw something very similar. So a little bit of staining in EBD68, lots of staining in echovirus 11, suggesting that echo 11 is just much more robustly inducing apoptosis in this system. So to conclude, human spinal cord organoids are a model for EBD68 infection of the human spinal cord. Post-2014 strains, but not the 2009 strain or the Freeman strain, are able to productively infect human spinal cord organoids. And this is reinforcing conclusions from other in vitro and in vivo models about the neurotropic potential of 2014 strains. And it's actually the first study that looks at the 2018 B3 strains in a CNS model, supporting what we all suspected based on the 2018 AFM season. And we learned that EBD68 induces apoptosis, but is less lytic than echo 11, which suggests to me that neuron loss in a patient is likely driven via a combination of viral damage and also the host immune response. So I have many people to acknowledge. I feel like I should always start with this slide so I don't run out of time, but lessons learned for next time. Thanks very much to both the members of my lab and also of our AFM collaborative working group, which has been a really fantastic group to be a part of. Also to our funding and to the patients that have inspired this work. So thank you very much.
Lovely. These old strains, have they been passaged on some sort of inappropriate cell line, and that is what's driving the lack of infection in anything natural? What are they passed on?
So I can't speak to much of their history, but EBD68 is often passaged on either HeLa or RD cells. And I imagine that that's happened many, many times by the time I get a vial of Freeman from the ATCC in the year 2020.
Is there any reverse genetics for this that you can wind back the clock?
That's a terrific question. I would say that our reverse genetic system is somewhat in its infancy and has been fraught with some challenges, but there are multiple groups that are working on it, although I wouldn't say that it's robust and reliable at this time.
And then the last question is, do these things synapse in the organoid? Can you see anything which is...
So I don't know how to patch clamp an organoid.
But even just looking by, microscopy, can you see anything which is tantamount to a synapse?
I'm hoping that I can someday make a fluorescent virus so that I could do live cell. I think that would probably be really helpful as a way to look at that. But with what I can do now, I haven't been able to see anything that specifically looks at that, but I'm not sure that I've asked it in the best way.
Yeah, it's cool. It's a lovely model, Megan. It's lovely, really nice.
Bernardo?
I guess a little bit of a comment to the sequence, and then so I would say the reference sequences are not passaged extensively, but you have to get enough to grow it, right? So I will say that that's the best you can do, and then or compare from the clinical sample. But yeah, c'est la vie. I guess my question is, with the 3-disc model, do you know which of the 3 cell types are being infected? And I guess a little bit of an extension to that. I mean, can you treat them as a tissue and section them and get around some of the staining woes of staining organoids, because they're beautiful. They're just a pain in the butt to get the antibodies there.
I'll answer the second question first. We've run into a couple of like Goldilocks problems with sectioning in that, they're too small to be like traditionally tissue sectioned, but like too big to be able to whole stain. So the whole staining is what we've been able to do most successfully. We've tried a couple of different techniques for like stacking a ton of organoids in like a low melt auger situation and then sectioning the whole thing. And that's probably the most promising. But we ran into a lot of technical challenges trying to do that. And then the other question is, I can tell you that at least neurons are infected.
Megan, do you notice any difference in cell numbers between infected and uninfected organoids over time?
Terrific question, and not significantly different.
Fascinating.
Meg? Megan, have you done any electron microscopy to? No,
I have not.
Megan, it looked like there were two really hot strains in your time course there. Is there anything special about those two?
Well, they were hot enough that I often now use them as my favorite strains. But the interesting thing is, I would say all of the strains that infected organoids that I talked about were associated with, AFM circulating years, but there is enough genetic diversity across all of the clades that we've not been able to specifically say like, oh, this one has capsid mutations or something that has been able to point us specifically to mutations that would be driving a phenotype. Oh, it's a mess, Paul.
Yeah, I'm wondering, you did say that you kind of identified the pathway affected in the neuron death. Do you know anything about which proteins was upregulated or downregulated? And do you imagine some kind of treatment using that information?
Victoria, I'll have a much better answer for that in like 3 months. I'm working on it. Actually, the guy that you're sitting by is helping me with it, so. Well, thank you.
Yeah, Paul's going to help identify a receptor. Okay, it's a pleasure to introduce our next speaker to focus a little on the clinical side. Dr. Carlos Pardo-Villamizar is a professor of neurology at Johns Hopkins University. He completed medical school at Universidad Industrial de Santander, which is one of the top medical school and universities in Colombia. Then did neurology residency in Colombia and also at Hopkins, where he stayed and rose through the ranks, and is focused on neuroimmune diseases and neurotropic viruses. And that's what he's going to talk to us about. If do I have to switch slides, or do we have? Okay, click to exit. And there we go. Okay, there we go.
Fantastic. Wow. Thank you very much for the invitation to be in Pittsburgh. From adopted Baltimorean Ravens fan, it's great to be in the Steelers territory. So I am a neurologist, I am a neuropathologist, and one of my passions is to evaluate and investigate the interaction of viruses with the nervous system. And obviously, acute flaccid myelitis offered the opportunity to investigate that interaction. I'd like to acknowledge first that I don't have any disclosure that is important, but I'd like to acknowledge that I am part of the acute flaccid myelitis working group. Many clinician researchers got together back in 2018 and put together basically a multidisciplinary group that take a look of different aspects of acute flaccid myelitis. I invite you to take a look of the website that is listed there. There is also a YouTube channel that contains at least lecture from 2 different symposiums about AFM, and Megan, we got the great opportunity to listen to Megan in one of those symposiums as well. So I invite you to take a look because it's a lot of information, not only about the clinical aspect of the disease, but also about the epidemiology, virology, as well as immunology. But you already got introduction to acute flaccid myelitis. And again, for a lot of people, acute flaccid myelitis became the poliomyelitis of the 21st century. And it was really a challenge for many clinicians around the country, particularly after 2012, when outbreaks of acute flaccid myelitis were identified in California, Colorado, and after that in 2014, 2016, 2018. Fortunately, in the past 4 years, we haven't observed major outbreaks of AFM. It's very important to recognize that AFM is a disorder that happens in other countries as well. We have colleagues in Europe that have been tracking down outbreaks of AFM. Interestingly, there are areas of the world where there is not too much evidence of AFM outbreaks. However, I believe that the problem is mostly diagnosis and surveillance. And we need to be aware about that, but because it's very possible that this disease has been there for many decades, but we were able to recognize that disease only until 2012. And again, the common denominator of this is upper respiratory infection. The upper respiratory infection basically hits different family members, and one of those family members, particularly children, get affected by upper respiratory infection, and unfortunately, some of them may get symptoms of neurological disease that we call acute flaccid myelitis. The most interesting part of this disorder is it's frequently concentrated in young children, is not necessarily frequently seen in adults. That is actually one interesting commonality with other viral diseases. However, it's interesting because it's not common to what was observed in polio, where adults were affected as well. But again, I think that in adults, we need to improve surveillance for the interaction of viruses and presence of acute flaccid myelitis. So what is the problem from clinical point of view? We have an upper respiratory infection. We suspect the enterovirus D68 is the culprit of this. But the interesting part is there is only one major area of the central nervous system that is affected, that is the spinal cord, followed by some areas of the brainstem. So this brings a very interesting question from pathophysiology. Is this the virus that is selectively infecting neurons, or is this an autoimmune response, or is an immune response that is targeting specifically motor neuron pathways? I believe at this moment we haven't answered very clearly that question. So I know that there is pathology evidence, at least from one case, of viral disease, but I believe that we still have ignored the potential role of the immune system affecting the motor neuron pathway. And I will show you some elements from clinical information about that situation. So this is basically the outcome of patients with acute flaccid myelitis, is extensive damage of the motor neuron system that produce atrophy of upper extremities, lower extremities. And these patients basically are very similar to patients with polio, with extensive atrophy, and that unfortunately is very difficult to manage because we are not able to have a very good recovery of those muscle groups. The typical profile of these patients is as has been outlined before, is these are patients in which there is a prodromal illness, frequently upper respiratory infection, particularly presence of fever, presence of upper respiratory symptoms. And these patients, after a few days, develop more complications. Some of them noted nuchal rigidity, and immediately after that, there is a very fast progression of neurological symptoms, particularly with weakness, that may affect also part of face, producing facial paralysis that is occasionally sometimes bilateral and produce a lot of lower brainstem dysfunction. And one of the problems of this is that after these neurological problems is established, many of the patients actually develop very rapidly neurological problem that leads to respiratory dysfunction. And many of these patients, unfortunately, end up in the intensive care unit with respiratory failure. So this is the common denominator in the clinical profile of these patients. And the patient basically suffer a very prolonged clinical syndrome that require not only intensive care management during the acute phase, but these patients unfortunately need to go to acute rehabilitation unit where they remain for several weeks and perhaps months just in the process of a neurological and respiratory recovery. This is our experience in 2016 when we did an assessment of the patients that we evaluated at Johns Hopkins and the Kennedy Crick Institute. You can see in the blue bars, basically the temporal profile of prodromal illness, particularly respiratory symptoms, but you can see the profile of neurological problems in orange bars. So again, in matter of hours, these patients were paralyzed. Some of them actually have a more prolonged course of illness. But the most interesting observation in terms of the profile of this patient actually was outlined very well by our colleagues at the CDC, led by Adriana Lopez, who collected the information from all of that population that was affected back in 2018, during the outbreak of 2018. As you can see in this slide, that is the summary of that outbreak, you can see that the majority of patients had a prodromal illness that lasted an average, a median of 5 days. So 96% of those patients that were recorded by the CDC experienced that prodromal illness. And you can see that the evolution of the acute state of neurological problem was over 48 hours. So in other words, this is a very rapid progressive neurological disorder leading to paralysis and a lot of complications, including the respiratory problems. As you can see here in 2018, 60% of the patients had a very aggressive disease and require ventilatory support. Some of them, many of them, almost a quarter of them were quadriplegic when they basically were managed in the intensive care unit. So this is a very aggressive disease. And the aftermath of this actually is very difficult for many families, many parents, and for the children in general, because they are left with significant neurological disability that requires neurological rehabilitation. And after this, obviously, an aftermath of many complications and problems. So this is the urgency of understanding this disorder, because it's producing a quite devastating disease. Now, what are the most common clinical presentations? This slide that was basically this table that was put together by the Acute Flaccid Myelitis group when we were working in outlining the consensus for management, describe very well that the majority of these patients actually develop upper respiratory symptomatology, prodromal disease. Almost 85 to 95% of the patients experience that problem. But what I like to point out here is a couple of things. This is the common denominator of a motor neuron disease. Basically, patients develop weakness in upper or lower extremities and develop what is very classical, a flaccid phenomenology. In other words, these patients are completely floppy and they have a classical syndrome of lower motor neuron syndrome. And importantly, these patients actually have a quite important amount of abnormalities in the spinal fluid. The spinal fluid is typically inflammatory, although not all of them have inflammation. Almost 85 to 95% of these patients have a very prominent pleocytosis and increase of inflammatory markers in the spinal fluid. But the other important feature is between 35–45% of these patients actually they present also with evidence of what we call bulbar involvement or brainstem involvement. This is the reason many of these patients actually end up in ventilatory support, because they are not able to generate basically cranial nerve function, and they are not able to generate a very good respiratory control that lead later to problems with respiration and end up in ventilators. So it's really important to take a look of when patients come to the ER, how to do an assessment of these patients. Obviously, the evidence of weakness, the assessment of weakness, characterization if the weakness is associated with a lower motor neuron syndrome or upper motor neuron syndrome, and characterizing the temporal profile of these patients is very critical. And for the clinicians, it's extremely challenging in the emergency department or intensive care unit in the first 24 hours of evaluation to determine if patient has acute flaccid myelitis versus other type of pathology. So that's one of the reasons I think that one of the critical elements of the first 24 hours when we are evaluating these patients is to characterize very well the clinical features of the patients. And I always use this slide because I always say the diagnosis of a disease is equivalent to an equation, right? And you need to have different elements in that equation. The first part is to take a look of the age of the patient, to take a look of the profile of evolution of clinical symptoms, and to characterize clinically and with your testing tools, how is the presentation of the problem? So in other words, two elements that are critical for diagnosis is the patient profile, the temporal evolution of the symptoms, and obviously with MRI and other tools like clinical tools to evaluate the topography of those lesions. So this is the question that you need to have in that setting. And I am bringing that topic because when clinicians are dealing with acute flaccid myelitis, they need to think that acute flaccid myelitis is part of a syndrome that is acute flaccid paralysis, that is completely wide in terms of differential diagnosis. In other words, when we are dealing with weakness, acute weakness in a pediatric population, we need to keep in mind that there are other disorders that may produce the same spectrum of clinical symptoms. Let me give you an example. This is the spectrum of spinal cord disorders that may lead to weakness. Obviously, acute flaccid myelitis is here, but also we have other potential problems like neuromyelitis optica, like myelitis associated with other infections, or we may have another autoimmune mediated disorder that is called myelin oligodendrocyte glycoprotein antibody-associated disorder, MOGAD, that may produce identical symptomatology, identical clinical signs, and the clinicians need to be aware about that in the differential diagnosis. And unfortunately, on top of that, there are peripheral nerve disorders like Guillain-Barré, that is a disorder that is very common and affect children as well, and produce a similar clinical profile of weakness and some sensory abnormalities. And on top of that, there are other infectious disorders like botulism that once in a while is present in our pediatric patients, and myasthenia gravis, or even strokes of the spinal cord. So the clinician need to be very alert and analyze very well those patients to make sure that the focus on myelitis versus peripheral nerve disease is clarified. Let me give you an example of our experience at Johns Hopkins. So between 2010 and 2018, we did an assessment of our patients with myelopathies. And actually, if you take a look at this slide, you can see that during that period, we have almost 30% of patients with acute flaccid myelitis in that group. So that means that we need to be aware because there are other diagnoses like vascular myelopathies, like autoimmune disorders that may come to the equation. Now, I'm going to jump to the last comment is there are criteria for establishing the diagnosis of acute flaccid myelitis, and that actually was published in Lancet in 2021. Many members of the acute flaccid myelitis group were involved in that project. And I invite you basically to take a look of that paper because that outlined clearly the characteristic of those patients. I really ran out of time. I probably talked too much, but if you give me 1 minute, I want to show you some of the MRI features. Did I run out of time? Okay, give me a couple of minutes. So MRI findings are very helpful because give us clues about the involvement of the brain, involvement of the spinal cord. Again, in the brain, we have the opportunity to see brainstem involvement. That is frequently in areas of the dorsal region of the pons. The spinal cord definitely is affected, as I mentioned before, and is selectively affecting the anterior horn of the spinal cord. And you can see that very clearly in MRIs of this patient population. The wide matter tracts are rarely affected, and that is going to help us a lot. But the other thing that I like to point out is that there are other minor brain changes that you can see in MRIs. And I'd like to show you this interesting MRI that we published this recently in one of our multi-center studies. Is there are areas of the brain that disclose clearly that areas of the network of the upper motor neuron system is affected. For example, in this patient that had AFM, you can see that there was MRI evidence that the thalamic nuclei that is connected the motor system with the cortex is affected. So it's very interesting from pathogenic point of view, because that suggests, wait a minute, that means that the virus is going directly there? And I will say, I'm not sure, but it's very possible that the glial reaction or the physiological changes that happened in the motor neuron system is affecting those pathways. So in other words, if you find evidence of brain abnormalities like this, don't be surprised in AFM, because it's not only and exclusively a disorder that is affecting the spinal cord. Thank you. Sorry, I passed my 20 minutes limit.
Questions for Carlos? I'll ask one first while you're going there. Anyway. So, you know, the rapid onset of the neurologic and progression of the neurologic symptoms, do you think that gives a clue to the pathogenesis?
I think it's telling us something. I'm still a little bit concerned about giving too much guilt to the virus producing cytolysis of motor neurons. I believe that either antibodies or T cells may be associated in some degree to that type of pathogenesis. We have a lot of pleocytosis. And if we have pleocytosis, it's obviously because the virus has been brewing there in the central nervous system. So I think it's a mix. What we need to understand is we need to understand clearly is how the virus is interacting with the immune system to generate this massive reaction in the motor neuron system that is very symmetric, by the way, right? It's not as patchy as other type of infections. But again, I think that unfortunately, the only way to learn about that and understand that is having opportunity to do more neuroimaging and obviously more patients and examining that evidence and see if what is the main feature. And unfortunately, the only way also to understand that is neuropathological studies that allow us to understand what is going on with those other cells.
You did show that there is like multiple causes of myelitis, including acute flaccid myelitis, including Guillain-Barré syndrome. Do you think that there is some kind of susceptibility to get AFM because of genetic background or race or something?
Yeah, it's very likely that the host of the viral infection is playing a lot of role in that, and the immune system, the susceptibility to the infection. I mean, it's the same that we are dealing with many of the other viral infections. Why it's so a small group of patients develop these acute neurological disorders?
So I know one of the diagnostic criteria for AFM is having gray matter lesions on MRI, but do you ever see white matter lesions? And can you comment on your experience there?
I think that we can see white matter lesions, but when you see white matter lesions, you need to be very careful because you need to go back and make sure that you rule out disorders like neuromyelitis optica, but particularly disorders like MOG. MOG, actually in the past 4 years that we have been basically quite alert to detect cases of AFM, the majority of patients has been MOG. and I believe that the clinician need to be very alert to that possibility.
Carlos, I'm completely out of my element here, but when you look at the MRIs, are they different for kids that are quadriplegic versus the upper paralysis versus the lower paralysis?
So the distribution of the lesions actually is quite interesting. Most of the cases with AFM have cervical cord involvement, but some of them have the entire spinal cord affected. So there are different flavors. And actually we have been able to detect also in some patients that there are areas that are focal areas, not as diffuse as these cases that we are publishing, that basically have a monoparesis or only one limb that is affected. So there is some degree of variability. And on top of that, there are some viruses, for example, Coxsackie may have similar features plus other areas of involvement of the central nervous system.
On the point you just made, Carlos, about Coxsackie and other viruses, do we know what the MRI findings of polio would be? Like, I mean, the polio cases that still occur in the world are in places where MRI is not readily available. So do we know anything about neuroimaging findings associated with paralytic poliomyelitis?
There are few case reports, I believe, from India that show that. But again, I believe that there are some similarities. The pathology of polio is quite interesting because as a neuropathologist, I was curious to go back to the archives that we have from early 20th century at Hopkins, and actually it's a mix of a gray matter and white matter inflammation. Yeah, so that's the only thing I can say.
Well, all we have to do is let the anti-vaxxers have their way a little longer, and sadly, we won't be able to answer that question. Okay, join me in thanking Carlos, please. Yeah, as a virologist, whenever anybody asks me about seasonality, I just say, next question. Well, I'm really thrilled to introduce our keynote speaker, Dr. Peter Salk. Dr. Salk is a professor in the Department of Infectious Diseases and Microbiology here in the University of Pittsburgh School of Public Health, and serves as president of the Jonas Salk Legacy Foundation. Peter did his undergraduate education at Harvard and medical school at Hopkins, another Hopkins connection, and then trained in internal medicine in University Hospitals of Cleveland. He has spent decades doing research at the Salk Institute in cancer immunotherapy, autoimmune diseases, viral diseases, and vaccines, of course, and has done a lot of work in vaccines, and actually was very involved with bringing AIDS treatment programs to Africa, highly relevant to vaccines and global health. And so we're delighted to have Peter visit us in his second home here, weather not quite as nice as Southern California, sorry, to talk to us about lessons learned from vaccination against polio. Peter, welcome.
Thank you, John. Thank you, everyone. This has been a really interesting day so far. And what I would like to do is just to make some contributions from a different set of dimensions, so to speak, as some background in which all of this ultimately may end up being embedded. So let me make sure I'm doing the right thing with this equipment. 1 second. Okay, laser pointer. There we are. Okay, well, let me go on. We've seen this slide already, and this is just to say, this is a prelude to say that, yes, this is polio is an old disease. It's been with us for a long time and primarily was an endemic illness. And it was only with the onset of improved sanitation that we began to see epidemics. So it's sort of, it's counterintuitive in a way. Improved sanitation, infectious disease, that things should be better. But the situation with polio was that in the endemic environment with poor sanitation, infants, young children were exposed to the virus, which was prevalent in the community at the time when they were protected by maternal antibodies from placenta and breast milk. With improved sanitation, with less of the virus in the environment, the early exposure didn't happen while they were protected. It came after the protection had faded, and then is what the time that we have we're seeing epidemics. We've seen this photograph as well. People were terrified. A few of us may be old enough to remember what the situation was like. And there was great need for a vaccine intervention. My father, on the right of this screen, worked with Thomas Francis Jr., first when he was in medical school and then at the University of Michigan in Ann Arbor, both of them interested in the prospects of creating a non-infectious or an inactivated vaccine against viral diseases. The dogma at the time was you can't do that. They did work together on the first influenza vaccine that was introduced at the end of World War II. Following that, my father wanted to move into his own situation, came to the University of Pittsburgh, where he had a lab initially in the basement, and starting in 1947. This was a year before the School of Public Health was formed and was located in this other side of Municipal Hospital, which is now Salk Hall with additional building in front of it. He gathered a very strong team of co-workers, Jim Lewis, Julie Youngner, Byron Bennett, Val Baisley, and other people that were just extremely skilled and devoted to the work that they were doing on characterizing the poliovirus in a number of types, early work on vaccine. The general concept was that the virus in an immunologically inexperienced person would enter, grow in the pharynx and the intestinal tract, go through the bloodstream to the central nervous system, kill neurons, resulting in paralysis. Antibody would develop later on, but too late to prevent that transgression into the nervous system. Virus would be excreted in the feces. In contrast, an immunologically experienced person having had a natural infection or theoretically intentional immunization, the virus, when coming into the system, would cause a secondary immune response, a memory response. Antibody would come up early and block the bloodstream portal to get to the nervous system. So that was the fundamental concept behind the work that was done. One major factor that had to be dealt with in creating a vaccine that would be non-infectious would be how can you kill it in such a way that you can predict that you are not going to have any remaining live virus. This is an inactivation curve plotted on an ordinary arithmetic scale. You can see the decrease in the amount of live virus in the presence of a 1 to 4,000 solution of formaldehyde under the conditions shown. If you replot that same curve on a semi-logarithmic scale, you get a straight line. You can only measure in the lab down to a certain point when there still is virus left, but the prediction would be that if the reaction continues at the same rate, you can take it long enough to the point when you're going to end up with a single infectious unit per 1 trillion doses of vaccine. So that gives you an ability to control your inactivation procedure and make sure that things are going to be safe. It depends on the conditions. If you have a larger amount of formaldehyde concentration, 1 to 4,000, the inactivation goes more quickly than at half the concentration. If you have the wrong conditions, that straight line persists. It occurs at body temperature. At body temperature, 37 degrees. Drop it down to room temperature. The curve the inactivation proceeds in a nonlinear fashion on the semi-log scale. Down at 0 degrees, under the wrong conditions, not just temperature, you will not get a straight line. So the whole premise was, if you're going to do this, you have to be able to have a straight line inactivation to make sure that you're doing things safely. My father kept all sorts of things. These were original tube, or vials containing just such an inactivation experiment. So this is the Mahoney type 1 strain. On the left, 14 hours, 25.5 hours, 38 hours, 45.5 hours. So it's just really quite something to hold these materials in your hand and recognize these came from this experiment, this experiment. These materials were waiting for Bernardo to to get in gear. They're about to go through the CDC to make sure that no living virus is present before they come to the University of Pittsburgh, among other materials that were donated by the family to the university. So we're looking forward to Bernardo to working with you to get this done. And these are just some of the original vaccine lots that were used in the experiments in the early 1950s. This is the Watson Home for Crippled Children, which is outside, or will hopefully it's still outside of Pittsburgh. I think it may be slated for destruction at some point soon. These are where the original human experiments took place with the experimental polio vaccine. After initial work in animals, it was time to move into humans. That was done in the summer of 1952. And the idea was this is a group of kids who have already had polio. We can do initial experiments there safely because if you choose the right strain corresponding to the strain that caused their illness, what we're looking for is an increase. Will this experimental vaccine preparation boost antibody levels? But these kids would be protected in case there was anything that had gone wrong in the process. After initial success, then the experiments were extended out to expose kids to inactivated virus to which they had not been previously seen. Another institution, Polk State School, was engaged as well. All told, there were about 160 kids in these very first trials, but the antibody responses were good, and it was clear based on that model of protecting the transit through the bloodstream to the nervous system that this approach was absolutely likely to succeed. A next step then was to expand the trials into kids in the Pittsburgh school system. Before doing that, this was the time when my father and the National Foundation for Infantile Paralysis, which was supporting this work, that this is the time for your family to get immunized. This was the second such injection. The first took place in our kitchen 2 weeks before that. And one aspect was demonstrating his own confidence in what he was doing. The second thing, and again, I credit my mother for this, I believe, was wanting the kids, we want our kids to be protected. There are similar shots of my next two brothers. I was 9 at the time. The next one down was 6, and the next one down was 3, or 2, almost 3 when this took place. This is one of the first trials in the Pittsburgh schools. There were about 7,500 kids involved in those, and the kids and their parents are heroes in all of this, having come forward despite the fears what might happen. We want to be part of this initial set of studies to define how put this vaccine together in such a way that it can be used in a large scale trial, which was done next. And this is the National Foundation supported a 1.8 million child trial starting in April of 1954, last until the data were finally analyzed and the results presented in 1955. So these were the so-called polio pioneers, but we really can't forget the pioneers, the pre-pioneers in the Pittsburgh school system. So this was April 12th, 1955, which coincidentally or not was the 10th anniversary of the death of Franklin Roosevelt, who purportedly had polio. Whether this was Guillain-Barré, whether it was polio, one does not know, but I think that it was not unlikely that his paralysis was due to poliomyelitis. Tommy Francis, wherever he is there, had been my father's mentor, and he coincidentally was the person who was responsible for the analysis of the results of this major field trial. When the results were announced that the vaccine was safe, effective, and potent, pandemonium broke out. This was such an emotionally challenging disease, and that now there was something that had been demonstrated to work was just beyond in a sense. And these are just reporters reaching to grab the printout of the press release that was made available. The vaccine was licensed for use that same evening. I mean, if you can think of what life is like now, This is really quite remarkable. Kids were lined up around the country to get their vaccines. It took me quite some time to notice that this Smithsonian photograph has two lines of kids in Memphis in April of 1955. So you have a white line and a black African-American line. Just a little bit of footnote of our own history. Tragedy. The Cutter incident. The vaccine used in the field trials was unbelievably carefully vetted. The two manufacturers, Park Davis, Eli Lilly, had to demonstrate that they could make 11 consecutive lots of vaccine without any residual virus in it, monitored both in my father's lab and also by the government. After that, when the vaccine was licensed, other manufacturers had been engaged, one of which was Cutter. Sorry. Cutter did not really want to follow the instructions with respect to monitoring this linear inactivation. They had ended up with live virus in numerous of their. Okay, thank you. Whether water will help, we'll find out. Thank you so much. Did not report their experience to the government. They only sent in lots that did not have live virus in their own experience. Their filtration system was not really up to par. They would allow the virus to sit in non-filtered material with the monkey kidney cells still present, protecting the virus from the formaldehyde ultimately. So this was just a real tragedy. There were about over 100,000 kids ended up receiving vaccine that had live virus in it. A couple of 100,000 people altogether ended up being infected. There were 169 cases of paralysis amongst them and 10 deaths. So this was just a horrible experience. On the regulatory side, it tightened up everything. The government changed their procedures, their monitoring, reporting requirements, et cetera. Cutter went out of the vaccine business, but this was just a terrible experience to have happened. It never happened again. Everything now is been done absolutely appropriately. Next step was Sabin. Albert Sabin was not a believer in inactivated vaccines. He thought that it was absolutely necessary to have a live vaccine. It took a longer time for him to complete the work on his own, administering it to children. The large scale testing could not be done in the United States in the late 50s, early 60s. Went to Russia to do larger scale experiments. And ultimately, the vaccine ended up being approved for use in this country. One of Sabin's contentions was that the live vaccine would induce mucosal immunity, where the inactivated vaccine would not. And he was absolutely right. If you look at the yellow, here's the IgA nasal and duodenal with the inactivated vaccine, nothing. With the live vaccine, yes, you have both nasal IgG, IgA, and duodenal IgA. So absolutely right on as far as that was concerned. Looking at the results, this is the total cases of polio, paralytic polio with a dash line, inactivated vaccine introduced. Within 7 years, there was a 97% reduction in the incidence of polio in the United States compared to the average of 4 years before that. The oral vaccine was then introduced in 1961 at this point where there were about 3% left to go with a lot of hoopla. I won't go through all of this, and forgive me if emotions appear in my voice. I can't entirely help it. Whatever. The oral vaccine ended up replacing the injected vaccine, and we'll have more to say on that matter. You do the same trick here and make a semi-log graph. And what we end up with is the vaccine introduced, in 1955, the killed vaccine, and basically, essentially a straight line decline. The live vaccine was introduced here, no change in slope. And again, the hoopla was this is going to be a miracle. It's going to change everything. What? So there's what we have. The problem that had concerned my father is absolutely there, which was reversion of the virus, of the attenuated virus to virulence. And from the beginning, one saw live virus vaccine associated cases. This is later abbreviated to VAPP, vaccine associated paralytic polio, which continued at around 8 to 12 cases a year, as long as the oral vaccine was used. But it was supposed to be a miracle. This is Sweden and Finland as a contrasting example, which only used an inactivated, a killed vaccine. Within 12 years, poliovirus was gone in that country. You could not even detect it in the sewage. So hoopla on the one side, on the other side, in at least that environment, the inactivated vaccine was able to eliminate polio entirely. The oral vaccine was the vaccine of choice for use in a global program to control and eliminate poliovirus. Let me see where I am here. Less expensive, easy to administer. You don't need medically trained personnel to put drops in a baby's in a child's mouth. I'm going to show a few snapshots of the progress of this. Before any vaccines, there were around 600,000 cases a year, approximately, of polio. By 1988, when the Global Polio Eradication Initiative was undertaken, with preceding and earlier work from Rotary, been a hero as part of this whole process. By 1988, we're down to 125 countries with persistent polio. In 2006, this is part of the snapshot, we're down to remaining 4 countries with endemic polio: India, Pakistan, Afghanistan, and Nigeria. It was thought that India would never be able to control polio, just given its circumstances. But we'll get back to that in a moment. Graphing it in this form, you can see from the time that the program was inaugurated, a fairly rapid response in terms of reduction of polio globally, but then a plateau. And for the next 10 years, there still was remaining polio without much in the way of progress. So another snapshot in 2012. By that point, India did flicker out. It did get polio under control. So we were left with the other 3 endemic countries and a couple of countries in which polio had, where there was a non-endemic transmission. New problem. CVDPV, circulating vaccine-derived poliovirus, polio caused by the circulating viruses that had regained virulence and were able to cause polio in the same way as the wild viruses. So there were outbreaks first detected in about 2000 with the 3 different types: type 1 immunologic types, type 1, type 2, and type 3 of the virus. Looking this piece of time up through 2014, these polio caused by the CVDPVs, primarily type 2. Okay. Almost nothing in the way of type 3, a little bit of type 1. Okay. So don't worry about the picture. This is starting what I've already said before, vaccines, about 600,000 cases of polio a year, 1988, about 350,000 cases, then starting to go year by year, starting in 2010. You can see the number of cases caused by wild virus globally reducing. There's a bump up around COVID. And then the red over here is the vaccine-associated paralytic polio, children receiving the vaccine or their immediate contacts, like mothers changing diapers and so on. It's really hard for me to dig out the good data here, but the best estimate early on was around 400 such cases a year, reducing somewhat with some changes in the program. The circulating vaccine-derived poliovirus-induced disease, getting down to 5 cases in 2016, but then there were some changes I'll discuss in a moment. It was recognized that we just could not continue to use the oral vaccine because of the polio that was being associated with revertant virus. There was the type 2 virus, which was causing most of that, was removed from the vaccine, theoretically under cover of introduction of the injected vaccine, which was never adequately done, plus add COVID to the mix, and there was quite a bump up. So looking at the percent of polio that's related to the oral vaccine itself, we're in the 99% range during the most recent period. So what do we do? Here's the absolutely current snapshot ending on the 24th of October this year in terms of cases within the red caused by the wild polioviruses, cases caused by the circulating vaccine-derived, the CVDPVs, primarily type 2, some type 1. And this is not counting the VAPP, the vaccine-associated cases in vaccine recipients themselves. The idea had been that, well, we were going to need to remove type 2 from the oral vaccine. Let's get IPV into the OPV only countries, dark blue countries using only, light blue countries using only IPV, about a quarter of the countries. Dark blue using both IPV and OPV, 10% of countries, and the rest of the white countries only using OPV. So roll out IPV into those white countries. Here's Nigeria getting IPV into the routine immunization system, OPV being used door to door and wherever one could reach people and as part of the global outreach. I'm going to break from that theme just to say that not only were there issues having to do with vaccines, and we'll get a little bit into those in a bit, we had all the social factors that had to be dealt with. 2004, rumors in Nigeria that the oral polio vaccine was contaminated with sterilizing agents to sterilize the Muslim population. That caused a cessation of vaccinations in part of Nigeria. Polio came back, spread across the continent. So this was one thing that was not very good. Pakistan, 2013, 14, 15, polio workers being targeted and killed, vaccinators. This a tough nut to crack, dealing with the cultural, religious, political differences that exist in the world. Problems with inaccessible children. This is Afghanistan, kids that couldn't be reached. This is with the Taliban, etc. By 2018, that had gone up to about 1 million kids that could not be reached by the oral polio vaccine drives, and the scale is going to change here, and that's what we just now saw. By 2019, off limits, larger parts of the country, so we were up to 10 million kids in July of 2019 that couldn't be reached by the vaccination programs. One thing that is coming in from another angle now, this is looking at the cases of polio in Afghanistan in 2018 caused by wild polio viruses, the original polio viruses not associated with the oral vaccine. That year, there were 7 cases of virus, let's put it this way. Of those, the total cases of polio caused by the wild polio viruses, 33%, 7 of them, 33% were in kids that had been fully immunized with OPV. That means 7 or more doses of OPV. I'm sorry, I'm going to bring this other angle. OPV, the savior of the world, best in all different parameters. What's going on? Something's going on in the intestinal tract. I don't have a full handle on it, whether this is malnutrition enteropathy, whether this is competing enteroviruses or other intestinal viruses, unknown. But the fact of the matter is, in some parts of the world, we saw what happened in Sweden and Finland. That was not a problem, but at least in this part of the world, there are kids that do not respond well to the oral administration of the vaccine. That had been preceded by observations back in 1978, same situation in Nigeria. This is the percent of immunized kids lacking antibody against either type 1, type 2, or type 3. With the live poliovirus, after 3 doses of the oral vaccine, half of the kids lacked antibody to type 1 and type 3 polio virus. The inactivated vaccines, you didn't see that problem. So something about the oral vaccine in some parts of the world is just rendering it not fully effective. So it was chosen for good reasons, but there are problems with that vaccine. Okay, this is a study by Sutter and his group at WHO. All I want to really look at here is this. This is looking at the excretion of live virus after an OPV challenge in kids who were either naturally immune because of having had polio, kids who were vaccinated with OPV, or kids vaccinated with IPV. And there are a couple of more decimal points that could be added here. Basically, 99.99% there was a 99% reduction in viral excretion in the naturally immune or the OPV vaccinated children, and only 95% reduction in kids who were immunized with IPV. So it's absolutely correct. What we saw before with the IgA is borne out here, that the oral vaccine does a better job of preventing growth in the intestinal tract of polioviruses, whether it's vaccine viruses or the natural virus, and excretion of the virus to be passed on. Question mark is, with 95%, how much of a difference does that really make? It's less good, but is 95% maybe good enough? I was hoping that would be the case, but the answer was no. This is Israel in 2013. Wild poliovirus was detected in environmental samples and in kids, in the feces of kids who had been fully vaccinated with IPV only. The world had begun to make a change and to move away from OPV because of its dangers in the more affluent countries. So IPV only was being used in Israel. Nobody got paralyzed. There was the blockade. You had the antibodies in the bloodstream to prevent the virus from getting into the nervous system, but the virus was able to circulate. So in this era, when the issue is eradication, in some parts of the world, the injected vaccine is not going to do the job. So here we've got 2 vaccines. Each has some problems associated with it. And how does one do the juggling act? I'm just going to step back for a moment and look again at the paralytic polio in this country. And this is the VAPP, the vaccine-associated paralytic polio. So here's the reduction in the naturally occurring cases. But from the start to the finish, when finally in the year 2000, the United States had a couple of years ago made the decision that 2000 would be the end, no more oral vaccine in this country. But for that entire time, we continued to have ongoing cases of vaccine-associated polio in this country. And this is Vincent Rock in yellow. saying in 2015, we now understand that every recipient of OPV excretes within a few days viruses that are more neurovirulent than the vaccine strains. What one hears sort of the conversation is, well, it's very rare. You know, the reversion of the attenuated strains to virulence is a very rare event. It's a very rare event that people get paralyzed by those revertant strains, but it is not a rare event that reversion to greater neurovirulence takes place. This happens all the time. Sorry, anger comes up. There's been a new oral poliovirus vaccine created for this eradication endgame using codon de-optimization. The idea being, let's do it now with the modern tools that we have. Sabin did the best he could back in the day when that you couldn't do this. Now let's do it in a way so that we'll avoid reversion to virulence. This is a lot better. It's being tested. Unfortunately, there still is some leakage. There still is some reversion that takes place. It's a lot better than what we have been using, but it's not 100%. Okay, so here again, we are just dealing with a situation where it's just not 100%. It's not 100% clear how best to go about this process of completing the end game. I'm going to allow my emotions one more little, something or other, which is the following: the communicative accuracy standard. And this, Peter Sandman happened to have been a roommate of mine in the pre-college science program at Jackson Labs in Bar Harbor back in whenever it was pre-college. So he came back into my life with this misoversimplification. This is a play on words from a George Bushism, where he advised people not to misunderestimate him. This was an excerpt from the Global Polio Eradication Initiative 2011 training manual. Question: are there side effects from the vaccine, the oral vaccine? Answer: no, there are no side effects. Experience shows that if children develop diseases or symptoms after vaccination, it is a mere coincidence. They would have developed these diseases anyway, with or without vaccination. Okay, that doesn't feel so good. This is 2022. Global Polio Eradication Initiative. CVD-VPVs, questions asked and answers, and answered. What are CVD-PVs? A child cannot get CVD-PV infection from receiving a polio vaccine. That's quite true by definition. These are circulating vaccine-derived polioviruses. So here's fiction. It's fiction that a child can be paralyzed from receiving OPV. No, it's not. Sorry. This is now a UNICEF publication for something for the GPEI, also 2022. OPV does not cause polio. Why are we saying this? I mean, I know why this is being said. It's we don't want to scare people away from the vaccines, but is there a way of doing it that doesn't involve contrary to fact statements? And this is not an official statement, but it's just another one of those things. The way ideas are circulate. This is a Scientific American website from the conversation. This is after the New York polio case appeared in reviving questions about the live oral vaccine. This is 2022. Again, the statement: the weakened form of the live virus in the oral vaccine cannot cause disease. Okay, this is not an official publication, but there it is again. And then finally, this really got me. And again, remember that graph in Sweden and Finland, and the experience in the United States. The eradication of polio in the Americas, Europe, and Africa has been accomplished solely through the use of the live oral vaccine. Okay. So, model smallpox eradicated, done. Can we do it again? I hope we can do it again. It's possible that we can, but we've got a lot of complexities to juggle to get to that point. So this is sort of a crossover point now with this conference. These are data now as of October 24th of 2023. So this is not full year data. It's only through the end of October, looking at the number of cases of polio caused by wild poliovirus, by the CV DPVs. And one of the measures that's been used in terms of surveillance is how good is surveillance with respect to acute flaccid paralysis. And so here, I just can't quite do this right. Proudly shown, so to speak, are the AFP cases that, when detected, demonstrate the capacity of countries and regions to pick up cases of polio and et cetera. The full year data for 2022 was about 100,000 cases of AFP. And again, I just sort of react emotionally to this. Yes, it's really important to have that kind of measure, but what are we thinking about AFP? What are we thinking about the AFM segment of this? I mean, it's really a shame that it's great to be getting rid of polio. It's absolutely important, but we cannot forget the kids who are being affected by AFM and other causes of acute flaccid paralysis. Okay, gonna wind down. These are busts of my father and Albert Sabin included in the polio wall of fame at Roosevelt Warm Springs, which Franklin Roosevelt had set up as a therapeutic environment for children affected by polio, honoring those who did major work with respect to understanding and getting under control the polio virus. Two different men, two different personalities, two different ideas. At this point, it looks as though, at least for the moment, using both of these techniques is going to be important. If we can lay down a good basis of immunity with the inactivated vaccine and a routine immunization, supplement as needed with the oral vaccine, especially with the new vaccine that is less susceptible to reversion, and doing it and taking into account all of the sociological factors that need to be taken into account. Won't go into more detail on that. It's the best shot we have at this point. Many partners involved. This was in 2008, the 50th anniversary of the Polio Wall of Fame, inducting Rotary, WHO, CDC, UNICEF. Now there's also, of course, the Bill and Melinda Gates Foundation and Gavi, the Vaccine Alliance, that are part of this whole program. This is a big deal. It's taken a huge amount of international cooperation and funding. Hopefully, we're going to be able to get this job done and be beyond this. And now, you know, in this arena, what you all are doing and hoping to do is going to become a next big and important thing. I'm going to put a little tail coda on the end of the presentation and just say a few things about my father. After the polio vaccine work in his hands had been essentially completed, he turned his mind towards establishing a new institute for experimental medicine, which he wanted to have at the University of Pittsburgh. It didn't work out politically here. He turned his sights to California, in La Jolla specifically. Did establish an institute designed in collaboration by Louis Kahn. The two had a very close collaboration together. It has really turned into a very important research establishment doing work on fundamental biology in a wide range of areas: cancer, stem cells, regenerative medicine, plant biology, neurosciences, It's just become a really extraordinary institution. My father himself, while there, did work that had started at the University of Pittsburgh in terms of particularly immunological interactions with respect to cancer, autoimmune disease, using myelin, study with myelin basic protein in patients with multiple sclerosis. Later in his life, after his lab was had been closed, looking into prospects for controlling AIDS in seropositive individuals through the use of an inactivated immunogen, hoping that this would also be able to be used prophylactically. It's not shown here as work that was done to continue to improve the production of the inactivated vaccine and get an enhanced potency vaccine, which did take place. I was fortunate enough to be able to work with him on all of these projects in the 13 to 14 years I was at the Institute, working in his laboratory. Just this photograph just has so much heart to me. This was the remaining people in his laboratory team that had come with him from the University of Pittsburgh. Lorraine Friedman, his long time administrative assistant, Don Wegemer, Elsie Ward, Francis Urraco. I mean, these people stuck with him. And I personally had the great honor and pleasure to work with them in my own laboratory experience at the Institute. My father was interested not only in physical disease, so to speak, but he was interested in the broader issues confronting humanity. This was something that was always on his mind. He wrote 4 books: Man Unfolding, The Survival of the Wisest, World Population and Human Values, A New Reality, which he wrote with my brother. There's been a new edition that's come out called A New Reality, Human Evolution for a Sustainable Future. And finally, Anatomy of Reality, Merging of Intuition and Reason. What's going on amongst us as humans? What is our future? What can we do on a broader scale to deal with the kinds of problems that exist amongst us, not just on the physiological, infectious disease, chronic disease basis? We've got a lot going on. We just open the newspaper, and what are we going to be able to do to come to grips with and improve the nature of our human interactions? My father received the Jawaharlal Nehru Award for International Understanding in 1977 and gave this acceptance speech, which was called, "Are We Being Good Ancestors?" I think it's a perspective that we want to keep in mind as much as we can. And a quote drawn from that speech, my youngest brother, Jonathan, ended up identifying it and working with one of the original architects. After my father died in 1995, it was inscribed in the travertine in the entrance to the courtyard at the Salk Institute. "Hope lies in dreams, in imagination, and in the courage of those who dare to make dreams into reality." I think for all of us, we have our dreams. That's why we're here today. And we want to do whatever we can to solidify them into something that's going to make a difference in the world. And then just to hark back to my father and say that one thing that characterized him, at least in my mind, was his wisdom. And that's just a quality that he radiated in everything that he did. So thanks. I really appreciate being able to be part of this.
Peter, thank you so much. That was amazing, insightful, and really moving. I have questions, but I'll see if others have. I'll try and be a good host. Dr. Dermody. Dr. Dermody has a question. I can't imagine.
Peter, that was wonderful. Thank you.
Thanks,
Thank you
Terry.
for sharing so much of your father's life and of course, the history of polio and the vaccines. And, you know, I have to say the scenes of the children lining up for vaccination, 1955, not dissimilar to the lines that were all the way down this hallway for people to get into this room to get their COVID vaccines. That was, of course, January of 2021, so a different time than just 2.5 years later. I mean, it's really remarkable. You know, I was going to ask you a question about inactive poliovirus vaccine and eradication efforts, but I don't want to ask you that. I'll find you later to talk about that. But I'm really intrigued with the last part of your talk and your father's thoughts about the human condition, how we can improve it, what our legacy is, and whether we are good ancestors. And I'm curious what you would think he would say now. How can we be better ancestors than we are right now?
Okay, I have a real hard time, in a sense, with he thought about things in a really broad way, and what he felt was that we needed to be thinking about the right sorts of concepts. Actually, let me just give you an example of the way he thought about things. He used world population as a model for his thinking, and referring to the sigmoid or S-shaped curve that one sees in many different situations. Put a couple of fruit flies into a closed environment, the population growth will be exponential, and then it will plateau. You see that here in so many different ways, and that's what he saw taking place with respect to the human population, that we went through this enormous growth spurt, and we're at a point where it just couldn't continue at that rate. We are living on a finite planet. We don't want to see this kind of a growth curve. So ideally, what we're in the process of seeing is a transition to a more stable state. And what he thought was, okay, in this first phase, when the curve is shaped like this, he referred to that as epic A, and saw different values and ways of thinking associated with being in this part of our human evolutionary trajectory versus the kinds of values and thoughts and drives that would be present in this phase, which he referred to as this epic A and epic B. So he thought about this conceptually in terms of a necessary transition. And one of the things that preoccupies me, so this is taking it out of my father's realm and is just very personal to me in a sense, is how can we take advantage of some of our inner capacities to help smooth out this kind of transition? We're living in an era where there is a lot of stress. Are there ways in which we can use our own inner resources, our own consciousness, so to speak, to help quiet things down, to help create a more peaceful environment in a very mechanical way? And I think there are ways of going about doing that. And I think that we need to incorporate some of those. I'm not getting into specifics at this point, into our thinking. We have capacities to be loving, kind, compassionate, wise, sparing, cooperative, etc. How can we help to cultivate those qualities to help us get through this?
I have a somewhat more pedestrian question about the vaccine strategy, and you alluded to this. When I was a pediatric resident in the late 1990s, we were giving, and this, I believe it was CDC guidance, we would give, I think, 1 or maybe 2, but maybe it was just 1 dose of IPV before giving the OPV. So why does it have to be either or? It seems that would be the best of both worlds.
No, I think that's absolutely right. It's a simple solution.
So what are the barriers to that? Is it just the logistics, or is most of the eradication team, which I know is a huge multinational effort, are people sort of so wedded to OPV that they can't let it go?
Well, I think that is true, again, given the data from Israel, it can't be let go at this point if we're going to completely stop transmission in some parts of the world, whether this has to do with the northern climes and sanitation and pharyngeal spread as opposed to the fecal oral route. We're probably not going to be able to let it go, but I think getting IPV into the routine immunization system, starting it before OPV to protect against the vaccine-associated paralytic polio, even if that will still continue to be a problem with these novel OPVs. There are just logistical issues. It's more expensive. The supplies have been limited. Was a disincentive against ramping up production on the part of the manufacturing, the pharmaceutical companies for something where we're just on the verge of eradication, et cetera. So and there was really an interesting experimental vaccine being looked at, microneedle patches at Emory, I think it was. Sorry, my mind's blanking for a moment. And that couldn't be pursued because at the point at which it was ready to ramp up, IPV supplies were so limited by the need to get them out that there wasn't enough to do the experiments that would be necessary. So I don't have an answer to how this is going to be able to work. It needs to have sufficient resources, and there is the disincentive of, well, we're almost there. I wish I had the wisdom, but I don't. I don't have it.
Thank you. Okay, go ahead, Anna.
I was just curious about when you said that when it was announced that it worked, it was approved that night. And then what was the process like for getting the distribution of the vaccine across the country? Because there were issues with distribution for COVID, the COVID vaccine in the US. So how does it compare? Like, was there a lot more planning back then, or what did it look like back then?
My hearing is less good at this age than it was a time. So you're asking about distribution of the vaccine. This was an issue. The vaccine at that point was in the hands of individual physicians' offices, and that's the way the AMA wanted it to be. My father would have wanted to see it be distributed in a more accessible way, as we have just gone through the experience with COVID vaccines, but there was a lot of resistance to that. Then when the oral vaccine was introduced, somehow the transition was made to Sabin oral Sundays, SOS, making it widely available and so on. So it would have been better if it had been done more along the model that we've most recently seen. But even with that, we've done okay.
It's an interesting point, though, Anna, just because I'm thinking now of a different virus, of respiratory syncytial virus, right? There are now 2 vaccines for older adults and a maternal vaccine and a monoclonal antibody. They're all licensed this year, and they're all extremely hard to get, availability and distribution and so forth. So, in some sense, we're still struggling with the same things almost 70 years later. Okay, well, before we go to the panel, let's thank Peter one more time, please.